The award with Tacit actually comes back to the Rett Syndrome Genetic Summit that we had. When we asked about highlights from a scientific point of view, people were very impressed with the progress of RNA editing in general. In addition, everybody pretty much mentioned how surprised they were by David's talk and how advanced trans-splicing had come. Monica had reached out and invited them to the summit. They were a relatively unknown company. They're a very small company.
This is David Nelles. I'm co-founder and CEO of Tacit Therapeutics. We've been excited about Rett syndrome for a while. It's been on our radar. We've been curious about learning more.
There was a lot of buzz around what they had presented. It's a technology that RSRT had been interested in for a long time, but the development wasn't quite there. And I think that was something that the field had recognized as well. And in the last few years, trans-splicing has kind of taken off. And the audience at the Genetic Summit was struck by the efficiencies that Tacit was able to achieve in mouse models.
One of my jobs is to be optimist in chief. And I think over the past 2 years, that optimism has been increasingly backed up with an accumulation of data. There are already programs in clinical development in our field. There are a number of ways to repair mutated genes out there. One key difference with RNA trans-splicing specifically is the fact that we repurpose enzymes that are already present in human cells. We take machinery conducting normal processes linked to day-to-day, moment-to-moment behavior and repurpose them.
How do you bias away from what the cell normally wants to do to get it to repair mutations? The best way to do that is to use what evolution has already given us. I don't think going in we had a sense of how advanced they were in their top program. It is an approach that can work for almost all mutations with a single therapeutic. The other advantage to trans-splicing is that you don't have to worry about the overexpression. Too little, or you have Rett syndrome, and if you have too much, you have MECP2 duplication disease.
Goldilocks gene is one way to think about it. It can't be too much, it can't be too little.
What I think has changed is the technology and being able to screen tens of thousands of options.
Convergence of new technologies when it comes to computation, AI-driven design, and high-throughput screening.
And that is one of the things that's kind of opened the doors to making this technology into a drug.
We can engineer, create in a tube up to 100,000 individual molecules, place them on human cells, create a hierarchy from top to bottom among all 100,000 molecules, which ones are the best and which ones are the worst. And that gives us a couple sort of new directions. So it's really about the scale and the speed that we can go from choosing a target to building new designs. So we have an immense amount of optimism that these principles will apply to Rett syndrome. We've now created a system that's ready to apply to MECP2 and use that to very quickly make new therapies. And then if we take that and also combine it with the institutional expertise that's been developed at RSRT regarding delivery, safety, clinical development, and regulatory aspects, position us, I think, better than ever before to help children who have this devastating disease. RSRT is still clearly interested in advancing what's possible and doing even better for patients, and also being scrappy when it comes to thinking about how to bring in new creative solutions to tough problems. There's sort of a startup mentality there regarding a recognition of how things are and how things should be.
Several people had come to us and said, this was the best conference I've ever been to. Part of that was the science, and part of that was actually the interactions with patients and families.
It's so hard to talk about like what these parents went through in a way that doesn't seem trite and trivial. I just have a lot of respect and appreciation for the stories they brought regarding what these kids are going through. I think I'm really optimistic that we could go on an accelerated timeline and get this to patients much faster than what has been typical historically in gene therapy.